Okay, good afternoon. Is it afternoon here? Yeah, good afternoon, good evening, good morning to some of the regions that folks are joining us from. On behalf of Team Axiom, I extend a warm welcome to all of you, as well as Akash Tewari. Akash is joining us from Jefferies. Akash does not need a whole lot of introduction to this table, as well as some of the audience that are joining us here. Akash is coming to us from Jefferies. Before that, you started off at Evercore,
Yep.
Followed by Wolf Research, and then we have a lot of common shared contacts there as well.
Yeah, absolutely.
Yeah, and then so one interesting tidbit about you is that you were triple ranked. What does that mean?
There's just different sectors in II. That's it.
Okay. You're triple ranked, including number one in SMID cap biotech. So your coverage areas are large cap pharma, large cap biotech, SMID cap biotech, as well as specialty pharmaceuticals.
Right.
Okay, so folks who are joining in and who wants to work at investment banking on the research side, you can ask questions for Akash. But for the rest of the session here, we're going to actually refrain from discussing any stock performance or discussing any stock advice or any investment in any specific companies. Our job is to talk about science, unmet clinical needs, innovation, and solution geared towards patients. Any thoughts?
No, I'm looking forward to it.
Okay, thank you for coming in. I know it was a long ride coming in from New York City, and welcome to Princeton. It's a beautiful time during the holiday season.
It is. No, it's first time in Princeton, and I can see where the hype is.
Yeah, it is. It is quite a beautiful town, especially in the middle of the Christmas sessions here as well. And then the team is here, so if you have questions coming through, and if you're able to pick up the questions from the Q&A, feel free to jump in. We're going to keep this casual. So, Akash, you and I kind of started at the same time in IO back in 2015, 16. Looking back, what are some of the memories you have at that time on oncology, especially IO? What surprises you the most sitting in 2024?
Well, back in 2015, I was dealing with generic companies and Valiant. So I was pretty far removed from, the Keytruda Opdivo battle. But honestly, I think it's a great case study of how lucky we are, because think about it. When when that asset got in the hands of large cap pharma, did they know what they had? Not necessarily. In fact, if you looked at some of the early data on CTLA-4, it probably looked like the more attractive target. Pharma company was they were investing in 4-1BB pretty heavily at that time. And I don't think we could have predicted PD-1 inhibitors to be the breakthrough that they really were. Mm-hmm. I think it's also a little disappointing that there's been maybe not that second and third leg.
Sitting now, quarterbacking back.
Sitting now. It's also kind of interesting to see. I do think Keynote 189 was such a transformative trial, partially because the data was like their phase 2 data that was generated with chemo and PD-1. It was good, but Keynote 189 actually took a step forward in squamous, non-squamous populations, in KRAS. Like it was almost like a breakthrough that was moved the field 5 years ahead, and we were all trying to catch up. Mm-hmm. So I think it is one of the seminal breakthroughs, I think, in all of oncology. And it was remarkable to kind of see it play through. It's also kind of funny to see how Bristol, or let's just say biomarker-driven strategies of Ipi-Nivo, if they had maybe just run studies for a longer period of time and not been so hesitant to go TNB high, TNB low, perhaps there would have been a different standard of care in first-line NSCLC. I think it's, but certainly I think a remarkable breakthrough, and remarkable to see from a distance. I did not live through that on the ground level. I was dealing with generic shortages and all that.
Yeah, that was really a golden era. And then looking back now, I feel when are we going to see that era come back again? Like, Dr. Langer and others would joke, like, in the last phase of his career, in retirement phase, he's seeing all of these lung cancer studies, actually getting approval, showing positive results. So
right.
And I think just kind of like the Gleevix of CML.
Yeah,
right?
there's
When
not
are we
many
going to see
Gleevix.
after Dara and multiple myeloma? When are we going to see the next golden era in some of these areas? So that brings us to a point like drug development is not a exact science, right? There's a lot of art into it, and I think there's a lot of human brilliance that goes into creating a compound. Or some would say it's a little bit of a luck. I know you and I had a different different disagreement in terms of what we think how drug development should be a little bit more reasonable, maybe, right? So the reason I say that is like I was talking to a scientist, and he said like as an industry we tend to be very copycat-ish.
Yeah.
If somebody's developing ADCs, then 200 other companies just jump on it. I don't really like it, but it seems like you don't mind it. Why?
I mean, I've just grown. Minding and not minding, I think, is irrelevant. It's just the incentive structure. The best way to change that is basically to do what Europe does, which is once a drug gets approved in a certain target, you're not going to approve anything else unless there's some added benefit of care, right? That would be game over for a lot of fast follower companies that day.
Mm-hmm.
Is that a good idea? I don't know. Mm-hmm. If you didn't have TMD1, would you have Enhertu later?
Mm-hmm.
Right? I think, look, I think it's always, this is just my general view, is we should accept the chaos. It's good.
Yeah.
Innovation occurs in an environment of overinvestment and waste. How are you going to have innovation without, by definition, having failure? We're an industry that doesn't have the economies of scale of tech.
Yeah.
Right? We have fixed costs. We have, unfortunately, science does not operate in a linear manner. So how do we look ex post facto and tell you, what was the right investment and the wrong investment? And like the discussion we were having, I think there could be like, this is not in the context of oncology, but like, let's talk about A beta mAbs and Alzheimer's. 20 failed phase 3 trials, Aduhelm shows a signal. I think you could rightfully say that, maybe we were data mining and seeking a signal, but then you have 2 other studies that are starting to show signs of life there. And now there's probably going to be more iterative success in Alzheimer's. If we had given up on that target, would we have gotten to that place? And then, for our work, we're focused very much on preclinical Alzheimer's. Mm-hmm. So it's the same A beta drugs, but for patients who don't have cognitive decline, who have been living with plaque for the last 10 years. And if you remove the plaque in the right context with the same MOA, ARIA could be dramatically lower. Cognitive benefit could be quite higher. Mm-hmm. You could diagnose patients with p-tau217, so a blood-based biomarker, you're not doing a PET scan. Mm-hmm. And then that might be transformative in terms of the amount of patients who actually develop more severe dementia. Mm-hmm. So who was right?
Yeah.
Right? What was the right amount of waste with that MOA?
So maybe we need some kind of a guardrail, like what is a reasonable threshold of quote unquote waste?
I just think the innovation and the breakthroughs are sometimes so iterative. Like what was Enhertu? Mm-hmm. For years, it was not, you wanted to go MMAE payloads and you wanted to go with more cytotoxic payloads and you wanted to have higher internalization. And you didn't want greasy biomarkers because if you did, greasy bystander effect, because if you have permeability on a toxin that's MMAE, what are you going to get? Peripheral neuropathy. Mm-hmm. Okay, but then when you do that with a topo-based toxin and you suddenly go DAR8, you have actually really cool PK, mm-hmm. and you're able to saturate the tumor site, and you're able to have an effect in HER2 low. So, think about it. That's probably the second big breakthrough in oncology development is like these topo payloads. And now everyone's going, I'm sure we'll talk about how everyone's gone too far into topo, but who cares? Like, I think to me, we as a sector, this doesn't bother me. Like they've ranked what sectors are most hated. Yeah. We're lower than the government. And it's a sector.
I think we're lower than the tobacco industry as well. Isn't that true?
People need their Zyn. No, but I think we do such a poor job explaining what we do and why we do. Mm-hmm. And I don't think enough people actually go out and justify the investment that we have in this kind of like beautiful, in my opinion, beautiful public-private hybrid structure. We don't live in a command-based economy. Yeah. And so, yes, like Steve Rosenberg might do a lot of the work in the NCI and then it gets out licensed to a pharma company and there's a price that they have to pay. But that's the system that I think works best. And,
Yeah.
How do you decouple the quote unquote waste with the innovation? I don't think you can.
Mhm.
Right. So I actually think of America is that we do choose to spend a lot of our health
Mhm.
on patients who are in their last lines of care. I mean, as we're in the discussion about oncology, do you think we would have that discussion in other places? No, they just let, unfortunately, people pass away. Those treatment options aren't available to them. If America didn't invest in that, would we have the same drugs and the breakthroughs that we've had before? That's not a discussion people really want to have.
Yeah, it's true.
Right. So I think but it is kind of interesting. I think if you were to ask most Americans, do you want people to invest in drugs and technology? I think the answer is actually usually resoundingly yes.
Yeah.
And maybe there is fingers to blame within pharma, within hospitals, within other areas where there's more fixed costs. But I think we should champion what these companies are doing more than we do right now. At least that's my two.
Makes sense. So talking about investments a little bit, obviously we're going to go deep into oncology in a second here, but obesity, I actually was not a big believer 24 months ago. So I'm seeing more and more data. What I feel like it's going to be a very different sector in terms of HCP engagement, engaging with the patients. It's not going to be the same thing how we engage on other, other diseases that we generally treat in the Western world. What are your thoughts about the investments that are happening in obesity? Is it impacting the rest of the sector in any way? And how do you see the HCP and the patient engagement differing from, let's say, oncology or other lifestyle areas?
Yeah, well, it's interesting. I think people with the GLP-1 inhibitor, you probably have a much more personal relationship with that drug than other ones, right? Like if you're spending, let's say, $100 out of pocket for Manjaro, that's probably money you're allocating not for drugs, but maybe for takeout food, your Uber Eats order, or the gym membership. I mean, it's actually, it is a very different type of medication because it lowers your LDL, it improves your A1C, makes you feel better, makes you look better in the mirror, right? Like, so it just, I think it is by definition going to be more of a consumer type product. And I don't think we've really seen that. I think Botox is probably, there's probably similarities between the relationship people have with Botox and the relationship that they might have with a GLP-1 inhibitor. I think if you step back, how many diseases, the funny thing is with the RFK stuff, started to listen to Callie and Casey Means and RFK. And the funny thing is 70% of the stuff they say is probably stuff we'd all agree with. And then it's
good on the right side of the
end. But it is, I think one thing is true is the axis of hormonal metabolic dysfunction probably does lead to a lot of different diseases downstream. And so is it shocking that certain obesity related cancers, if you control your weight and you reduce your inflammation, that you're not actually at the same risk of developing those cancers downstream? No. So I think we're going to look back at these GLP-1 drugs as the first anti-aging drugs. And I think actually that's where the industry needs to go. This is the discussion we were having earlier. As much as it's easy to blame the finger at companies, but we're going where the river takes us.
Yeah.
Build a dam, right? I really would love IP and patent structures to change to allow companies to explore preventative therapy and giving them the financial incentive to do that.
Yeah.
Right? What if cancer vaccines don't work in a metastatic setting, but they work in an earlier stage setting? If you're, BRCA positive and there's a cancer vaccine that takes 10 years to run that trial to show that your incidence of developing metastatic breast cancer gets reduced 50%. Right now, the incentive structure in our industry doesn't exist to run that study. With the GLP-1 inhibitors, that's the beautiful thing is you're putting so much money into the space that they can run an Alzheimer's study for GLP-1s. Or you can run a study looking at whether cancers are occurring downstream. But you don't have that opportunity for a lot of other drug targets. I think we should work on changing that incentive structure. I think it's time because, again, I think there is a fair critique of we spend so much, and are we getting maybe the same lifestyle, the lifespan extension that other developed countries are getting? well, by the way, if you remove, unfortunately, drug overdose and suicides, the answer is yes, we're actually in line with every other developed country. But my point is, like, let's create that incentive structure to occur, just like with the orphan drug law, just like with Hatch-Waxman. Those were just seminal, bills that actually an entire industry was based off of. I think there is room for more innovation on the legislative side. That's what I think that would actually have a big impact on the amount of people who are developing some of these more severe cancers.
Good. I'll pick up on the targeted question that you had in terms of targeting. Back in the day, I think it was in the section of GBM. I was at an ESMO in Amsterdam or Copenhagen. I remember listening to the speaker. They were saying that they didn't really see value of an IO and IO combination in the GBM setting, but more of an IO and a VEGF, right? So that was like bringing you back to the VEGF discussion we were having earlier.
Yeah.
And now we are seeing a lot of movement that way. What are your thoughts? And we're starting off with lung, as you can tell.
Yeah, I mean, actually, I think it's really, like, so VEGF is an active target across multiple tumor types. PD-1 is an active target across multiple tumor types. If you improve the therapeutic window maybe 30%, you might go from having a PFS benefit to having an OS benefit. This is not high science to a certain extent. The other thing that, like, we should also start thinking about the PD-1 CTLA-4 bispecifics that certain pharma companies have. Again, known targets, but, whether it's the avidity benefit, whether it's like modulating T-cell exhaustion, you might see the same targets show very different results. 4-1BB, right? There's companies that are doing bispecifics there, and there's emerging data that maybe spacing of the dosing could actually lead to, better outcomes. I mean, that's very early, it's uncontrolled. But my point is, like, these are iterative breakthroughs that can actually lead to big clinical benefit. Now, on the PD-1 VEGF stuff, I think the secret sauce really is the cooperative binding. I think if you also look at some of the transactions that have occurred there since then, that is what I think will make these drugs have a better therapeutic window. I think there are reasons to be very skeptical. I think there are reasons to be optimistic. The skeptical reasons, I think everyone knows, like, okay, VEGF, do Asian patients do slightly better? Can we read across there? Number two, VEGF, is infamously has a very weird relationship with PFS benefit and then actually OS. The curve separated so quickly. Would that occur with other trials? Did Keytruda underperform modestly there? Blah, blah. Like, great.
Yeah.
So I'm skeptical, and yet I actually lean a little more optimistic. And here's why. In the context of NSCLC. First line. Second line is a different story.
Yeah.
They are already showing something unusual. Because if you look at LiPo-7, if you look at Empower 150, if you look at traditional studies where you take VEGF TKI or BEV, combination with the PD-1 inhibitor, what do you see very quickly? Down doses, dose discontinuations. Right? It's funny, like, people, like, in our team, we've tried to model, like, the PK of one of these regimens versus the other. And I'm like, look, or PD-1 versus PD-1 versus PD-L1 is, if there's a difference. Does it matter when the comps were basically patients were discontinuing one of the arms or down dosing to a rate where it wasn't clinically meaningful? That's the point. So if you look at Harmony 2, at 8.5 months, there are no drug-related discontinuations on the IVON arm, ivonescimab or whatever. And that, to me, is the unusual signal, because there are, like other studies, double the amount of grade 3 adverse events. But there's no grade 4 or 5 events. And there's no treatment-related discontinuations. So then you ask yourself, well, what is the nuance between a lab-mediated discontinuation and a clinical site-mediated, discontinuation? I'm sure if you talk to those companies, they'd probably say, like, we're just keeping the patients on the drug. Like, yes, they might have lab-assessed hypertension or proteinuria, but they're staying on the medication because they can handle it. And if that actually leads to a survival benefit, that's interesting. So, like, a lot of people will say, well, Akash, you can't run that trial in the US or in Europe because patients would get lab assessed. But I think if you were MD Anderson and you said, hey, if Harmony 2 does hit on OS, are they going to say, I can't run that trial in the US because I would take Avastin off traditionally? Or would they say, maybe this is a good idea to keep the patient on the drug, educate the site investigators to handle hypertension And if you can stay on the drug for a longer period of time, then you're probably more likely to have a closer relationship between PFS and OS.
Mm-hmm.
That's the point, right? Like, because again, VEGF, like you're impacting, the blood supply, and once you get off of it, it's going to return. So you have to have sustained VEGF inhibition.
Mm-hmm.
So that's what's unusual here. Because like if you look at LEAP-07, the median follow-up was 15 months. HARMONY-2 was 8.5 months. If you look at the fireside I hosted, or like I had a KOL call with the guy who was the PI for the Empower 150 study. The big question is, at 8.5 months, the AEs you see there, is that representative of what it is 15 months in, 20 months in? If the answer is yes, then that is a really interesting signal and it's worth exploring. Now, the question is like, really, how big could this be? How small could it be, right? Like if you look at the TNBC data that came out recently, in PD-1 low patients, definitely a better signal. PD-1 high patients, not as clear, right? So like maybe TNBC, the breakthrough is going to be in the PD-1 low population. Maybe in, first line, it'll be non-squamous, and it's squamous. There's more AEs that historically show up with VEGF inhibition. So maybe it won't be used there. But I think anyone who pretends like they know how big this signal is going to be is lying. And I think if you were to look at the companies and the companies that have entered the space and the way they're thinking about it, this is going to be an arms race.
Mm-hmm.
They're running 10 phase 3 trials on with the TROP2 ADC.
Mm-hmm.
How many phase 3 studies are going to be run here?
Yeah.
And I cover companies that are in RCC, and it's basically VEGF TKI and a PD-1 inhibitor.
Mm-hmm.
Why wouldn't this be a place you could take it there? What about bladder cancer combined with PADCEV?
Yeah.
What about small cell combined with the B7H4 or a DLL3, right? Like there are areas where this is headed that I don't think are being discussed right now. And again, like, do we look and say there shouldn't be more companies developing PD-1 and VEGFs, or do we say let it go?
Yeah, but then to your point, if it's an organized chaos, right? What would you suggest? What are your advice to folks that will jump on this bandwagon now?
Nothing. I would say let the chaos occur, because again, if Merck did not have the development program they had, and also a lot of, or let's just say other companies that were developing PD-1 inhibitors, a lot of that was externalized R&D because mid-cap companies combined with the pharma. But if you didn't do that, would you have the adjuvant studies that are reading out with Pembro right now?
Yeah.
So what was the right or wrong amount of investment?
Yeah.
And like like when TIGIT will fail or IDO fails, like the companies die. Capital gets reallocated.
Survival of the fetus.
Right. So like, but what I would say is, this is why I think investing in oncology is so difficult right now, is because there's been a few fundamental changes in the space. One of them has been China, and I definitely want to talk about that. But the other one is like, if you're a pharma company, what is your structural advantage? It is not drug discovery. It is not necessarily making better drugs than other companies either. Your structural advantage is access to capital, to have brilliant clinical affairs teams, and the ability to have relationships with huge sites, enroll studies very quickly. And it is your ability to sell and market drugs at a global scale. It's not making drugs, right? So here's kind of the point. These companies, if you look at what happened with TROP2 ADCs, if you look at what happened with KRAS inhibitors, is the first gen KRAS inhibitors going to win the space, or are they going to be the ones that are 2 years behind? If you're 2-3 years behind and you're a pharma company, you can catch up pretty easily, and not to mention, you can actually learn from the failures of your peers in terms of biomarkers.
That happens a lot, right?
That happens all the time.
Yeah.
So the question is, why do you have to overpay for the lead asset? Right. So I think that's been the one of the fundamental issues with oncology. Can you and I, like, what have been the great, and I guess we can't talk about stocks, but like, what have been the great deals in oncology?
Yeah, let's not talk about stocks. But I'm checking if there's any questions.
But it's but my point would be, in oncology in particular, China's also changed the game here.
Yeah, let's bring up China a little bit. As you were discussing the VEGF and the PD-1, back of my head, I was because that back of my head, I was thinking, most of the data is generated out of China, right? So it seems like there has been a sea change on how we think about Chinese data and how are we bridging it to the European or Western population.
I mean, I think there's some real strategic advantages to China. Like I was talking to a company and they had, this new CEO came and fired his chief medical officer. And I said, well, why'd you fire him? And he said, well, we were running a trial and he gave me all these fancy sites in the US and I got a bunch of fifth line patients who had tried every single thing else. And it took forever to enroll. And I wasn't actually really able to determine quickly whether this was a real drug or not.
Yeah.
In HER2, look at their clinical development. A lot of it was run in China. Like, remember when that deal got announced, everyone's like, where did this asset come from? But I think a lot of people are replicating that model. If you run trials in China, you can get, there's some great sites there, frankly. You know, and whether it's, you can talk about the first BCMA CAR T that had a breakthrough. That data originally came from China. People were skeptical until now they're not. But you can run more naive patients. You can enroll those trials more quickly. You can use that data as a part of your clinical package. It can't be your entire clinical package, but it can be a meaningful part of it. You can understand PK/PD. You can dose, you know, up very quickly. And there's also, you don't have to release that data either. So you can actually make a lot of those decisions probably under CDA and have informational asymmetries that you can really use to your advantage. Think about it. There are, if you think about one of the companies that are running TROP2 ADC trials, and you're thinking about why are they running 10 studies and why are they in these weird biomarkers? Well, they have data in China that's not been published that's informing that decision. So why do I need to run these studies in other areas? What about gene therapy and like where maybe it's easier to enroll patients and get site access? So I think China's fundamentally changed the game. But then number two, if you think about maybe some of the pricing concerns from that domestic market, there are probably companies that are very willing to in-license assets to U.S. companies at favorable financial terms. And so if I was a pharma BD person, I'd be going to China all the time. And it has changed because, again, these companies can consistently catch up. A 2-year advantage is not that big of a deal, right? So it also means that if you are cooking up something that's just as good, any pharma competitive intelligence team is going to say, why am I going to overpay for that early asset? And that's that's a trend that's happening in oncology. That's also a trend that's happening in the obesity space. The guys who are leaders are agnostic. They're betting on everything. Right. So if you continually iterate on your lead product, you are also neutering your competition because you continue to build your commercial advantage, your payer advantage, your all the outcome studies you're reading out. But then when you're not just betting on one target and you're making your own internally, it actually makes it very difficult for people to catch up. That's how you create a moat. Right. And I think the other thing we're seeing with oncology is the moats that are getting created are often CapEx, right? Like whether it's radioligands or CAR T, like having the infrastructure and the payer relationships to actually sell some of these more complicated products are probably more formidable moats than science. And so I think it's just, again, so like, we're not irrational. Things are changing. But, again, the organized chaos, you just have to learn to accept it and love it, frankly.
So in terms of the big pharma or the mid-cap pharma, it seems like before you go shopping for biotechs in Boston and other areas, you might want to just go to China at this time.
I mean, how much of the science in quote unquote Boston is iterative? Let's be honest, a lot of it is. And a lot of those assets are basically in license from China, and then they put a new management team in place, and now it's a U.S. centric company. So I maybe feel a little. I'm sure there are going to be issues with. There are going to be certain sites where you can't trust the data, and there's certain sites where the data integrity and are they getting on the right dose? Crossover, what are they getting on relevant regimens? I mean, that's certainly relevant when it comes to the PD-1 VEGF discussion in China, but it doesn't mean you should be ignoring this information. And again, I don't think it's even worth debating now because look at the BD that's occurred from pharma companies over the last 2 years. It's the proof's in the pudding. If they trust it, why are we not?
You have a question?
So when you think about China, right? Obviously we can't ignore it. Do you kind of view China as, are they kind of taking the fast follower approach? Are they kind of going after more novel targets, maybe biosimilars? How do you kind of segment the Chinese market?
I think, I mean, you guys have been in ASCO over the last couple of years.
Yes.
What are the drugs everyone talks about? Often they're Chinese assets. Like,
Yeah.
So again, I think the line between fast follower and like iterative, I mean, what's not iterative at this point?
It's fair.
Especially when it comes to oncology. So I don't know if there's a meaningful difference. If there is, I haven't seen it yet. But I think, again, I think so much of oncology is once there's a breakthrough on KRAS, okay, you can drug KRAS GC12. Great, but the first agents were drugging it and hitting the target maybe 60, 70%. And then now you're seeing data coming out from other companies where it's almost like complete inhibition and you're not seeing the same mutations form. In fact, it's almost like a TKI where it's like, it's flat. But that's iterative, but led to kind of a breakthrough. Scemblix, CML, right? I mean, it was Gleevec should have been the be all end all, but it's allosteric selective inhibition that's more potent, and you actually have less mutations form, but you also have a higher MMR response. So, and some of the like again, a lot of the quote unquote improvements here are actually pretty clinically meaningful, but they're iterative in terms of the targets known. It's improving PK/PD. It's improving, the druggability of these assets. But
so there's a question coming in. I think the questioner is trying to get to like what kind of technologies or maybe modalities China is focusing on. So like what kind of therapies are coming out of China if you were to put them in buckets between novel MOAs, biosimilars, similar modalities, but new targets like ADCs. So maybe trying to understand the thinking of how Chinese developers are thinking of the future and where they want to invest.
I don't know if I have a good answer on that, because I think if I knew that, it would be I would have to access to the private markets. Because the stuff that's public is mostly iterative and biosimilar. And I don't have access to the private stuff. But I just think proof is in the pudding, because if you talk to the pharma BD guys, they're going there more than ever. If you talk to VCs, they're going there more than ever, not just to raise money, but actually to find assets themselves. But I would probably say I haven't heard. Most of the stuff I'm seeing are known targets, but have better druggability in some aspect. It might be a novel toxin, it might be better PK, it might be a slightly better therapeutic window, it might be higher, levels of inhibition. I don't know if there's a new target that's come out that they've been per se in the lead in, but I don't think you have to be that in oncology.
There's huge unmet need. I mean, so in terms of lung, what are you excited about for 2025?
It's okay. I mean, I think the PD-1 VEGFs, I think is super interesting. I am interested in that. The PD-1 CTLA-4s. So IO bispecifics or PD-1 VEGF bispecifics. We should probably start looking at PD-1 TIGIT, though I'm not a big TIGIT guy. But I think we should generally be thinking about if a drug that like single agent activity, improving the therapeutic window, can we revisit known targets and suddenly think about them in a slightly different manner? I am kind of interested in like spacing out 4-1BB dosing. And I think, again, like it is a broader discussion we should have is like, are we overdosing Pembro? I think we, frankly, I think there's probably evidence that we are, but do we really think about T-cell exhaustion sequencing therapies? I think that's super important for blood cancers like multiple myeloma, but I think it's probably going to be relevant here too. I'm going to sound stupid here, but I haven't given up on cancer vaccines in a metastatic setting either.
Why not?
I think it's about, so this is my janky theory. I don't know if it's true, but has there been probably proof point that if you resect a tumor that a cancer vaccine can probably prevent disease-free recurrence in certain TNB high populations, and even actually in pancreatic? I think we're seeing the answer is like, yes, that's probably true. So what if you can combine, let's say, a PD-1 CTLA-4, or sorry, a PD-1 VEGF combined with a TROP2 ADC, combined with a cancer vaccine in lung cancer? Why is that interesting? Well, like you have off-the-shelf regimens that will debulk the tumor. And then the cancer vaccine might take a year.
Mhm.
Like, so it won't even have a benefit on ORR. It may not have a benefit on PFS, but if you look at like Dendreon,
Yep.
cancer, like, it showed a benefit on OS.
Mhm.
So if you can debulk a tumor with
Mhm.
off-the-shelf therapies and then induce
Mhm.
an antigen-specific response, maybe with an off-the-shelf, not a personalized cancer vaccine.
Yeah.
What if you and the safety profile looks benign?
Mhm.
Could that actually lead to an overall survival benefit?
Mhm.
I think the jury's still open there, right? So, if it's safe enough, every single company is going to suddenly start adding cancer vaccines to their arms. They're going to be running triplet therapies, right? So I think there's going to be some proof points over the next couple years there that I'm probably going to be interested in. What happens with the Trop-2 ADCs, right? Like they've been such a dud, but again, like squamous, non-squamous, efficacy, PD-1 refractory, non-PD-1 refractory, you're seeing differences in these agents.
Yeah.
And one of these companies is making a massive bet that there is differentiation in the space, and we're all ignoring it. So could there be certain biomarker populations where you're showing a greater effect size, and could there be nuances even between topotoxins in terms of where patients respond? I think also the biomarker-driven approach in first line. I mean, no one was happy with the second line data with Dato, but the biomarker-specific data was intriguing. You can't dismiss it. And so could you, with Trop-2 internalization, could there be a space in first line with that? Again, very unlikely.
Yeah.
But it's not, I wouldn't be dismissive of it. I think also like certain areas like KRAS, can you, because again, with the first gen ones, I'm like, you look at the Keynote 189 data in KRAS mutated patients, it was the exact same efficacy. It's like, why the heck? That would have been an example of like, this is waste where you're going after a targeted population, but you don't actually have better efficacy. Why the heck are we running that study? Fine. But if you're actually getting meaningfully better efficacy in that population, great. I'd love to see that play out.
So given if it's not a homogeneous disease, right, lung cancer as a whole, so it seems like you want to be a leader in lung cancer, you need multiple modalities in your bucket. You can't just have a PD-1 or just a KRAS.
Yeah.
What does that look like for a leader who wants to be a thoracic leader?
Again, it's a large cap game. There's probably 3 companies that can really do it at scale. 4. And I think even those companies may partner on some of those assets because the cost is just going to be so high.
Yeah.
But yeah, I mean, unfortunately, that's probably a sad part about oncology development. It's like you're basically, I used to, have the PD-1 branded. Now it's like I have the PD-1 branded, I have the Trop-2 branded. So I'm now charging $500,000 per patient for a 2-month OS benefit in a biomarker-driven population. Is that where first line NSCLC is right now? It could be.
Yeah.
So yeah, it's turned into a large cap pharma's game. Very hard to compete without sequencing therapies together. And I think if you talk to BioNTech and Uber, even if you talk to my dad, like my dad's an oncologist and a surgeon, when you talk to people who are treating, not idiots like me, the cure to cancer is complicated, and it's not Gleevec. There's no like one drug that's just going to change everything. It's sequencing therapies together. It's priming T cells. I think we should be thinking about that a bit more. Like even you can make an argument with PARP inhibitors in certain types of cancers. You get on a PARP inhibitor, you're probably likely to develop a very aggressive mutation afterwards. You might actually have worse disease once you progress up a PARP. Is it a good idea, even if they had a better data in first line or second line, to use them as aggressively as we do? We just think about indication, line of therapy, and efficacy. But I think there is probably a broader discussion to be had about like, well, what's the right way to sequence these therapies? Probably the biggest case study of that's going to be multiple myeloma, right? But I think there's probably going to be fair discussions about that in NSCLC. Another one where I think it's going to really hit a wall is HR positive breast cancer and like the overlap between HER2 low and HR positive.
Yeah.
When do you start using an HER2? Because there's 75% overlap between HR positive and HER2 low. So do you use an HER2 second line? Do you use it after endocrine therapy? Like, I think these are probably where the puck is headed. I think there's going to be some really interesting discussions. The last thing I would say is so many of our data sets are being generated in non-PD-1 refractory patients, but perioperative and adjuvant use of PD-1 in first line is becoming a bigger deal. So like if you're looking at data in head and neck and you're like, oh, this is showing really good efficacy. Yeah, but like if head and neck PD-1 inhibitors start to get used in an adjuvant setting, then we should really only be looking at data in PD-1 refractory or post PD-1 patients. And are we thinking about that as biotech investors correctly? That's kind of an interesting question. So I think the problem is 2025, and probably this decade's not going to answer any of these questions. Definitively.
That's very positive.
I think it's true. I think we're only going to get iterative answers. I think we're going to probably figure out over the next couple of years whether TROP2 does actually have a place in first line, given the toxicity that it always occurs. Right. I think we're going to understand in first line, at least PD-1 high on the PD-1 VEGF stuff, whether there's a real signal. I think we're going to start to know whether KRAS has a strong signal that justifies first line usage. Those are probably the quote unquote breakthroughs we're going to get in NSCLC over the next couple of years. I don't know if there's a lot.
So the decade might be more of an incremental and some sequential, smaller.
An increasingly complicated stratification in NSCLC in particular, right? Mm-hmm. Second line, post-EGFR, squamous, non-squamous, there's going to be that battle.
Yeah.
I just think it's going to get more complicated, not less complicated.
Makes sense. Let's talk about complications. So in hematology, I'm a solid tumor guy, but we talked a lot about multiple myeloma and CML.
Sure.
That whole segment, what incremental needs are there that still needs to be addressed, do you think?
I think one of the most shocking data sets I've ever seen in my life was Blenrep.
Like, that's the first topic on my agenda here.
I don't know. But I think if you can create a better Blenrep without some of the eye toxicity, or if you can improve on that and actually get better efficacy, that's a big deal. Like no one, I think that data set was so shocking, and it came out of nowhere because the trials weren't showing a really strong signal, and then suddenly you're having this big OS benefit. There, in my opinion, there's an open question of what do you do second line?
Yeah.
Right. And I also will say, with the T-cell engagers, which I think everyone is generally, no one knows what to do with. Some companies think they're going to be huge. Some companies don't. Are you showing a durable signal with BCMA CAR-Ts in first line? I think that's still up for debate. Mm-hmm. So I think multiple myeloma is really, really difficult because there's a lot of intriguing data sets and there's no, I think, definitive answers. But I do worry that maybe we're so quick to think it's going to be BCMA CAR-T first line. Mm-hmm. I think if you can take Blenrep and you can combine it with another agent in first line, you get very close to a BCMA CAR-T efficacy. The question is is it worth exhausting your T cells first line? I don't know. Is it worth using a T cell engager if you're not going to get a response to a CAR-T afterwards? Probably not. But there are going to be companies who are going to be pushing that forward. So it's I think that space is very much chaos right now. But I really do think Blenrep is probably, that data set is probably one of the most underappreciated. And if you can iteratively add on Blenrep to another off-the-shelf agent or make a better Blenrep with a wider therapeutic window, I mean, I think that would be a really interesting area for investment. If I was starting a company, I would try to, in multiple myeloma, I would try working on even ADC combos. We've been starting to see that too. I think that could be quite intriguing.
Question for you in terms of, we were in the area of cell therapy, right? So cell therapy in solid tumors, any specific approaches that may work? Go crazy on that one.
I don't think I can. I don't know.
I know.
Is there one?
Anything promising that jumps out at you?
You tell me. Has there been one that stood out to you?
No, not yet.
Not yet, right? I hope I'm not crazy.
No.
Even with the TCR stuff. No, I don't think I've seen anything. I don't think I've seen anything that's really a breakthrough, frankly.
Good. Okay, so let's continue on to solid. Anything else on him? We're not going to see another Gleevec in this.
I mean, I think, look, another thing that's again, I think this concept of T-cell exhaustion is incredibly important.
Yeah.
We keep forgetting, look at the CD20, CD3s, they're all beautiful data sets. But now, as you go to a year plus, the infections are really starting to increase. And that's damning. I mean, from a clinical experience, from how do we think about the infection rates with some of these bispecific engagers, and whether we should really be using them in second line as much as we are. And I think it's really interesting does a CAR T have that same level of immune exhaustion? I don't think do we see the same level of infection risk with CAR Ts? I don't think we do. So I think I'm actually getting incrementally more bullish on the CAR Ts, and I'm getting more and more skeptical with some of the bispecific engagers simply because of the infection risk that shows up and then how those patients progress once they've been on those treatments. I don't think it's talked about enough. And that's not just for multiple myeloma, that's frankly for DLBCL as well.
Cool.
Akash, can I ask you a question about the cell therapy? What's your opinion on the allogeneic CAR Ts that are currently becoming more developed?
I'm probably more bullish for them in the setting of I&I than I am. I don't know. I don't think the problem is the data sets we've seen so far I don't have the same durability as off the shelf. I still need to do conditioning, which is like, I think the big problem, because you also increase infection risk or the risk of developing T-cell malignancies. Talk about it for BCMA CAR T as well. So I don't know if they're meaningfully more efficacious than off-the-shelf therapies. So I haven't seen, at least in oncology, a clear, this is the right application. I think it would be quite interesting in certain areas in I&I, lupus, or some of these more severe autoimmune diseases where maybe that would be the right approach. But I don't know. I don't think I've seen it really show a killer data set yet in oncology. And correct me if I'm wrong. I don't cover any of those companies, right, or those areas right now.
Okay, so you touched a little bit on breast cancer. We were in the early days of defining HER2 low. How low was low enough? Right. So excited to see a lot of advances did take place. But it feels like there's still a lot unknown and a lot of things that still need to be addressed. What are some of your thoughts? Is there a differentiated way to approach breast cancer? Again, that's another disease. That's not
another large cap game.
Yeah,
another large cap game.
Because of the way
and it's so much money.
Yeah.
Actually, I'm pretty excited about HR positive, but I'll give you a hot take. I don't think this is a very consensus opinion. In my opinion, the most important drug in the development of HR positive breast cancer is going to be the CDK4s.
Yeah.
I think they've shown really, really intriguing data. I mean, if you're going to brag about Ribo showing 45% grade 3/4 neutropenia because Ibrance showed 60, you're showing combo data with fulvestrant where this is showing 15%. And not only that, but you're doubling the response rate with meaningfully lower grade 3/4 neutropenia. If you think about why Pallas failed and Abema and Verzenio caught up, it was like in a first-line adjuvant setting, are you able to have less AEs and more constant inhibition, better CBR that actually translates to PFS and OS benefits, right? That's the name of the game.
Yeah.
You are like, we did an internal PK analysis because a lot of people will say, well, Abema is already CDK4 biased. No. Some of the CDK4s that are being developed here are hitting CDK4 orders of magnitude more potently than the CDK4 biased CDK4/6s. Now, the big question is, are you going to need CDK6 on OS and durability of signal? We still don't know that right now. But man, I think that is that's a huge deal, because if there's anything that's going to work in first line, again, I think that is that is the most promising drug in my opinion in first line. And frankly, the combos you're seeing in second line are better than in HER2 low. Like in HER2 low, you're getting like 10, 11 months in that population. I get it. It's good data. But I frankly think you should be using in HER2 post, let's say, a CDK4 SERD combo. And look, SERDs, I'm not going to tell you they're going to work in wild type, but they do work in ESR1. And I think the beauty of what SERDs are being used now, and I think if you look at Serena-6 for certain European large cap pharma companies, what they're doing is really identifying where these could work. In my opinion, for the SERDs, the one variable I would love to see in all those studies is when did the patient get on a SERD after developing an ESR1 mutation? Because if you saw, this was in San Antonio a couple of years ago, the longer you were prior exposed to a CDK4/6, the better you actually ended up doing on a SERD. My suspicion is getting on a CDK4/6 actually primes you to develop an ESR1 mutation. And if you can get on a SERD, then you're going to have a really, really big clinical benefit. And so, certain companies are measuring ctDNA to find out exactly when it starts to drop or go up, and then they get them on these medications. So I think a CDK4 SERD combo in the right setting in second line is going to be better than in HER2 and in a HER2 low population, even though there's a huge overlap. So I'm super excited about that. I think some of the data we've seen with PIK3 alpha on top of doublet therapy showing a 3, 4, 5 month benefit is also kind of interesting. Again, stuff I've been skeptical on in the past, but the data is the data. So I think in first line, PIK3 alpha may have a place. I think CDK4 could be standard of care changing in first line, but I'm more confident on it in second line. So yeah, I think those are going to be. I think if you look at the studies that are being run, there's like 6, 7 phase 3 studies. I wouldn't be surprised to see standard of care change.
Okay.
I think I think things that I'm not as bullish on, even though there's some hype, CAT6, CDK2, salvage. Like if you look at the CAT6 data that came out, and I got a lot of attention because it showed 10 months on PFS, but if you look at the population that they enrolled, there was a 55% ESR1 mutation rate, and 91% of those patients were post CDK4/6. Those were literally the patients who would do incredibly well on a CDK4/6 SERD combo. Like a CDK4/6 SERD combo would have shown 15 months in that population. So why do I care about a CAT6 showing in a second, third line population 10 months with the neutropenia rates that they have? So I've never really got CDK2. I've never really gotten CAT6. I think they will be probably used fourth line after and HER2. I just don't think they're going to be game changers, in my opinion. Again, salvage setting probably.
Makes sense. So staying within the women's cancer setting.
Yeah.
And areas of gyne.
Right. Now, the last thing I'll say, and this is actually a broader discussion of like sequencing ADCs in general, and this is probably less on HR positive, it's more on HER2 expressing cancers. Why the heck would you take another HER2 ADC after you've already progressed on HER2? I just don't get it. But that's actually the answer pharma has, right? You're going to take MMAE ADCs after you progress on HER2 and then dose patients there. I actually think some of the data from ZW25, like a HER2 bispecific, where you're able to get patients off chemo as well, and you're showing longer durability of response, I think in combination with a PD-1 inhibitor, I think there's some real room to innovate in the post and HER2 space in HR positive.
Yeah. Do you have 10 more minutes to stay longer?
Yeah, sure.
Yeah. So the folks who have just joined in, so we'll go 10 minutes after the hour. So if you want to stay put. Okay, great. So let's move to RCC.
RCC, man. I don't know.
Sorry, RCC person. Each of us have a tumor, and so the RCC lead may be at the table.
I honestly think the most interesting study hasn't been announced yet, which is the PD-1 VEGF in that population.
Okay, so then,
right? If you look at like Lenvima Pembro versus Cabo Nivo, you could argue that Lenvima Pembro actually had better efficacy, but it was really good efficacy with the Checkmate 9ER regimen, along with the better quality of life, and then again, discontinuations and staying on therapy made a difference. So why wouldn't that be exactly where a PD-1 VEGF could really shine? I would, I would love to see that in combination with a HIF-2, right? But like HIF-2, I don't know. I don't think I've ever seen an OS benefit. I don't think I've ever seen anything very strong there. I think HIF-2, I'm not giving up on the target, but I think there's been recent studies with Zanza, Pembro, and they're probably going to combine with the HIF-2 in first-line RCC. Maybe HIF-2s have a place when you have VEGF inhibition and HIF-2 in the same context. Maybe that's what you really need. But I don't know if it's going to necessarily work. It's going to be PD-1 VEGF on its own. I'm skeptical on that. Those studies are reading out, obviously. But yeah, I mean, I think that's probably what's happening. And then, of course, with Zanza, there was a partnership with a large cap pharma. They probably data that they've seen that we haven't. Again, that's a shorter half-life VEGF TKI that hopefully can have a better therapeutic window in terms of patients can tolerate the drug, but theoretically you can probably get a higher Cmax. You might be able to get better efficacy. That would be incredibly interesting to see if you can improve meaningfully from Cabo and actually have a standard of care switch there. But yeah, I mean, those are the at least in my coverage. I'm sure there's stuff that I haven't looked at, but that's really where I'm focused on is kind of first line and second line, and
makes sense.
It's been basically the PD-1 VEGF stuff.
So moving on over to bladder prostate.
Wait, by the way, is there stuff that I'm wrong on the RT?
We'll let you know.
Oh,
It's Vian. Vian is not in today. Okay.
oh, okay.
She's listening in. Yeah. Vian, let us know.
Yeah.
So prostate cancer. I was actually in the room. Where was I? In Madrid.
For us.
For ESMO, is it? Yeah. So lutetium, the data that was presented, which is super exciting. Then the AR degraders and others. What are you excited about in the prostate? Don't be wrong. Your dad is listening in.
No, I am actually. I honestly don't think I'm an expert in prostate. To be honest with you, I think again, like there's a few companies that have all the assets and are sequencing stuff. I am interested in so the CYP inhibitors that there's a phase 3 study being run, really good efficacy, really bad tolerability. I don't know where that gets integrated in standard care, but it's really good efficacy. AR degraders in combination with radioligand.
Yeah. I
think Pluvicto combo with AR degrader could be really, really interesting.
Mm-hmm.
Again, I think there's some safety concerns with a few of those compounds, but again, I think safety is reasonable. I think there are actually probably some differences in PK/PD with some of those assets, and I wouldn't be surprised if some of the Pluvicto combos ended up being better.
Mm-hmm.
And then you've got just like this hodgepodge of like, non-androgen, salvage treatments, whether it's, Cabo being used in that population or next gen degraders. You've got the PSMA therapy.
Yep.
You got some really interesting bispecific data that came out, but again, how much of that was confirmed RECIST responses? We need more durability there. I don't know. If you were to ask me what is the breakthrough trial that's going to happen in the next few years, I'm not actually sure right now. But in terms of things that I'm tracking, AR degrader combined with radioligand.
Yeah.
I am tracking the PSMA ADCs. I'm tracking the B7H3 ADCs, which have been okay. I am tracking the CYPs simply because of the efficacy and if they can actually improve the therapeutic window. But I don't think anything's really emerged. And then the emerging PSMA masked CD3 data where you're showing really interesting efficacy. But again, there's a difference between PSMA response and then RECIST confirmed response, and I don't think we have enough durability there. So emerging, but I don't see the transformative therapy yet.
You mentioned bispecific. Do we need to relook or revisit bispecific at this time?
Sure. Look, again, T-cell exhaustion and the ability to have a sustained response. There's certain therapies where like DLL3.
Yeah.
Like, wow.
Yeah.
Remarkable data.
Yeah.
And again, for some reason, that target, you're having more durable responses. I think some of the early data that's been shown by some of these mass technologies, again, I don't know how that improves durability of response, but I think that's stuff to really think about. The funny thing is, like with these drugs, like I remember reading this paper for the what is it, epedorcatamab or whatever I'm just going to say Genmab, but it was there's CD20, CD3.
Got it.
And it was actually really interesting. If you read that paper and how they discovered the drug, they literally just took a bunch of constructs with different geometry and they just found one randomly that ended up having a better therapeutic window and better lower CRS, and somehow that worked. Like I think there's still a mystery about what's making some of these work particularly well and some that aren't. I did some work priorly on like IgM related CD3 degraders. And look, you could argue that maybe they didn't have the same potency, and that's probably true, especially for bulky disease like DLBCL. But it was really interesting that you were getting responses like 6 months, 8 months afterwards. You're almost getting pseudo progression. And you did decouple some of the CRS. And what if a more sustained T-cell exhausting bispecific approach makes better sense over time? I don't know. Like, so yeah, I think there's some really, I think there's emerging stuff that I cannot explain, that certain T-cell engagers seem to behave differently in terms of lower CRS, better durability of response, and lower infection rates. And that's particularly been in liquid tumors, but we're starting to see that show up in solid tumors as well. It's definitely exciting. The biggest breakthrough, in my opinion, hands down, is first line small cell with the DLL3 drugs. I mean, I think small cells probably the most transformative.
It has been a difficult one as well.
Yes,
right. In terms of, I think the diagnosis and early diagnosis has been, or testing has been helpful in a lot of segments, like breast cancer.
Not for prostate.
Not for prostate, like PSA has obviously.
I can't believe they removed. They told you not to do PSA.
Not to do PSA, right?
So people with more severe cancer.
Yeah, exactly. So where do you think there is still a lot of unmet need in terms of diagnosis, or not so much diagnosis, like testing earlier on, like even basic health screening that could actually help? And then that ties back to the question that's coming in. What advances or challenges are you watching in biomarker testing and patient identification technologies that it could be adopted in the marketplace in the next 3 to 5 years?
I think for the millennials in the room,
including
me, I think we're all like a little weirded out, like the rates of colorectal cancer are like dramatically going up. And
yes.
I don't know why.
But I know, like, I may talk to a lot of the peer group, and they're all getting colonoscopy at the hit of their birthday or whatever.
Yeah.
So the folks are screening.
Right. I mean, I think that, so is that because of increased screening?
I think so. I think you're seeing an algorithm where, like, folks, you wouldn't even you went for a colonoscopy? Like, okay, yes. Because maybe it's better screening at this time. I'm sure.
Right. Or is it, the food we're eating and the environment? Actually, that's probably the first one that comes to mind. I think there's been I think you want everything to start going where HR positive is for breast cancer, where people are getting identified earlier and earlier. I don't know if I advocate mastectomies as aggressively as maybe some people have them, but I would like to see other cancers get there. I think prostate cancer has taken a step back.
Yeah.
Because you've actually seen the opposite, where someone did some type of health analysis that a simple blood test like PSMA is actually not a good idea, and we're over-treating these patients. But they did that to the person who...
But that N=1 one that actually needed that. Right?
But
exactly.
Again, that's an example of are we helping or hurting by trying to be overly, do a lot? Apparently, it wasn't smart to have as aggressive PCM.
I mean, we've seen that in smoldering multiple myeloma. Like, folks, when we worked in it several years ago, peri-pandemic, folks didn't even think it was a disease state.
Right.
Nobody wanted to treat it. So here was great data that didn't get introduced because there was no solution that was requested.
Great point.
So maybe it's a time where we actually kind of have to retrain some of the clinicians that are working on it. It's like, maybe you don't want to wait and see, because that's not what we do here.
Yeah, I mean, you say that to the patient who actually does get the
right.
Exactly. They're not going to realize that nuance.
No, I've been in KOL calls and well, Mrs. Smith is coming in Monday morning at 9, and we're going to tell her, we're going to wait and see. And I'm like, I'm not sure if I'm going to be happy if I was Mrs. Smith, that you're just going to wait and see. What is the threshold and what is the acceptable threshold? Right?
Again, I'm probably more of an advocate for aggressive testing and doing it earlier. And I don't care if maybe some people might get overtreated in certain contexts. I think obviously there is a legitimate and important QLAI or whatever assessment depending on. But again, I think the fact that a lot of urologists are now backing away from holding off on PSMA testing, I think is an important case study. I think also legitimately, and I would give a lot of credit to my dad personally for this, is you have to think about communities, like for African American people. So he's done a remarkable job. He's created a bus that actually has an MRI and a PSMA test, and he's actually going to the communities, especially post COVID, where a lot of people just didn't get tested, and you've seen rates go high. But I think you have to use the tools and adapt them to the populations you have. And there are huge and legitimate health inequities in the United States that need to be addressed. And particularly for African American people, it's as simple as giving them a PSMA test aggressively. And of course, you're also seeing there are genetic disparities in terms of how aggressive someone's prostate cancer is.
We saw zip code disparities and around UPenn, how patients in different zip codes getting on a treatment and not.
Yeah, I mean, I'll say something a little controversial, but not funny, but the thing about America is you have the best healthcare system in the world, and then you'll have certain areas where you have maybe
access issues,
very substandard healthcare. But America's actually both at the same time. I think if you had any advanced disease, America's going to be the best treatment you're going to get in the world. It's actually consistently there. But it's actually separate countries. There's
having access to that. Yes.
Yeah. So I think particularly for prostate cancer, again, for the African American community, again, look at who's getting the severe disease. Don't be blind on that. Yeah. And I think sometimes, especially post-COVID, reaching out to the communities directly is critical. Because again, I don't think it's new tests per se. A lot of the tests are out there. It's just getting it to them.
Or maybe the next town hall we want to discuss about that. Yeah. So maybe we'll save off of that.
Yeah, I will say the last one I think would be kind of interesting. Like if you look at Gardasil, like there are next gen versions of Gardasil as you go to 14 valent, and like oral cancer, anal cancer, there are other cancers, GU cancers, where perhaps if you can increase the valencies, and if you can go single dose, again, not a lot of people ex-US can take 3 doses of Gardasil. It's hard. Yeah, I can't take 2 doses. Hard for me. I'm sure it's hard for anyone else. But I think that would also be really interesting, is if you could reduce some of those, the prevalence of those, just like how Gardasil was I think it's an amazing success story. If you can expand the valencies and get single dose, that again, from a utilitarian perspective, could have a transformative effect in terms of the incidence of certain cancers showing up long term. And again, if you think about benign, hyperplasia or whatever for prostate cancer, how many of that actually does lead to patients eventually developing real prostate cancer? I really think we need to create IP incentives to allow people to run 10-year studies. Wall Street does not operate on 10-year cycles. It doesn't operate on even 3-year cycles. It operates on 1-year cycles. But if you can, I have no problem giving a company 10 years, giving them a priority date 10 years back, if they are willing to run that trial, and if they can actually show with, let's say, a cancer vaccine just targeting, PSMA or some known target, that you actually develop prostate cancer at a severely lower rate. Why aren't we creating the incentive structure to run those trials? Right?
It makes sense.
But then we are totally willing. If you look at the Natalie study of I remember looking at ASCO 2 years ago, like they did an NNT analysis. If you take a branded CDK4, CD4K, and fulvestrant, like first line, it's like $600,000 to prevent disease recurrence. I don't know if that necessarily makes a lot of sense. Frankly, even if I look at the cancer vaccine data, so like if you look at the Nadina study, this came out at ASCO this year. If you take just 2 cycles of Ipi/Nivo, peri-adjuvant, you have an 85% event-free survival out 3 years. Why would I take a fancy cancer vaccine with Pembro for 3 years to have the exact same event-free survival? So I think it's often not necessarily new treatments. It's just using them in the right context and creating the financial incentive to adopt them. That could actually have a really transformative effect.
Yeah, and Cherry's giving us a fact check here is that increased CRC amongst young people is not believed to be by increased screening. So
there is something wrong.
There is something wrong.
It's a microplastic. I don't know what
Plastics, red meat in general, linked to it.
Microplastics are asbestos, basically.
It's very interesting. Let's talk a little bit. We can talk for days, but we're not going to do that because I know you have a lot of work to do. In terms of biotech, I always feel like biotechs have become the outsourced model for big pharmas, where a lot of the drug developments are done. Innovation is actually happening in a very agile setting. But the biotechs have gone through a very difficult time in the last 4 or 5 years. I'm in Boston. Coming out of Boston, you're depressed because you're sitting in front of a client. They can't even do the studies that they need to do because the Series C funding is not coming in, or the devaluation of their whole company, et cetera. So what could be done? Because the bigger ecosystem need the biotech to kind of have this symbiotic relationship for the biotechs to actually come back to homeostasis.
Yeah.
Is there an approach where we kind of need to relook at that biotechs are not independently on their own. We can't just let it falter and say, okay, because there's a lot of innovation that's going out the door. Let them burn. Okay.
No, but why not? I mean, that's the
away from the chaos theory, though.
No, I think you should. So my point is, why not? I mean, if drugs don't work, or if there isn't scientific, there isn't a strong enough scientific rationale to continue to invest in it, isn't that another way of looking at that? That's the logical and ethical thing to do, is to not continue to invest when there's no data.
Which is fine, but I think we're now at the tail end of a biotech bubble. The situation we had created is now adversely affecting. But
I think it's worth looking at, well, what created the bubble in the first place? And I think part of that is, again, like, look, you can't, if you're married to capitalism, you got to dance with the one who brought you. Okay. And there are going to be like our economy, usually, unless where the Fed is like continuing to just put money, it is cyclical.
Exactly. Yeah.
There will be periods of exuberance, and then there's going to be a course correction.
Yeah.
That's just human nature.
Yeah.
Right. I think the thing that happened in kind of 2020 was, I think a lot of life science funds looked at what was happening in the tech world, and the tech VC model completely changed, right? Historically, if you look at tech VC, 1 year out, you were down. If you gave $100, you were 70 cents. You were $70 in, because 3 of them failed. Maybe 1 or 2 of them got a Series B, but like, it's not like they were growing exponentially. Then you had certain funds who weren't tech VCs, but they said, basically, I did an analysis, and if I just invested in everything, as long as I captured the ones that hyper scaled, it would actually make sense for me to invest in all of these and do it at scale and speed, and not act like I'm some type of arbiter of what's good or wrong. Like, I'll give you the money, I'll give it to you without telling you what to do, and we'll see what happens. And the problem was, then suddenly the biggest value generation, sometimes privately, became, did you have the right funds invest in your Series A? Because that determined who invests in your Series B.
Yeah.
And then if you change how you got paid as a VC on paper gains, then you are creating value by simply funding companies. What does that lead to? It leads to a lot of companies that probably shouldn't have IPO'd.
Yeah.
And then if you do that in a life sciences, because there's fixed costs and science isn't linear, you're going to have a bunch of preclinical companies IPO. Okay, but does that mean we destroy the system? No, like it happened, right? Like what is the better system? I don't know, right? Like I just think why is biotech immune to the exuberance of the market that every other sector goes to? Right? If the .com bubble in the 2000s, all of those pets.com failed. That's not a good. So my point is, did we give up on? So how we say Silicon Valley shouldn't exist anymore?
So how long should this suffering phase continue for the biotech?
I think it's already had some really healthy corrections. I mean,
do you think it's about time? 18 months coming back?
No, I define coming back. Look, again, we need to get over, and this is what I kind of tell people, who cares what public markets are doing? If you have a good asset, IPO. If you have good data, IPO, because there's clearly demand and there's money on the sideline, as we all know. I actually think they're not as correlated as people think, and I think people overthink these things.
Yeah.
Because, and just to tell you, think about it. We had a really tough sector period in the XBI, while the greatest period of growth in the pharmaceutical industries was concentrated on 2 companies on the biggest market of all time. And at the same time, the XBA underperformed the broader sector. If that is not the biggest case study of decoupling of value creation in the sector, I don't know what is.
Yeah.
There's a company that was approached $1 trillion market cap for the first time in healthcare.
Yeah.
In a period where the XBI underperformed. If you look at the P&L, and this is actually where I think you have a great point in terms of, we aren't probably misallocating stuff. Again, if you did a P&L of what actually had the highest IRR and what we spend on, there's probably some misalignment. But the point is that people learn. The companies that I'm betting right now often have human data. Great, healthy. I like that. The companies that are IPOing have a better syndicate where you're taking chunkier, you're having institutional investors write bigger checks. And what I would argue is that it's very important, if I was a biotech CEO, to think about syndicate formation. You're looking at some of the companies that have actually not gone down when the market went down, it's because 70% of their float was tied up. Right. And I think that's actually what you're seeing with the VC innovation models. They're actually doubling down, and they're creating their own they're finding the asset, they're creating their own management team, they're incubating those companies, they're owning more privately and letting them, develop more so that they can own more of the economics when it goes public. But that company did the right thing. They got human data, they generated it, and it should do well in the public market, right? So I think, again, the great thing about capitalism is that we can't afford to be wrong for too long.
Yeah.
Unless you're a sell-side analyst, I guess. But even me.
You have a great rating.
But seriously, these companies have learned. I think and if you were to think about the companies that IPO'd, how many of them were doing crazy science that was unvalidated? Now, at the same time, you could come back and say, well, all these companies are iterative. It's just another IL-17. It's just another IL-23. It's a long-acting IL-4. But they're going to work, and pharma companies want it. Because if you also look at what pharma companies have started to buy, again, they are doubling down on the moats that they've actually established, which is commercial infrastructure. So the IRR on a drug that is a validated target, but they can put it into their portfolio is much higher than taking some of that scientific risk.
Yeah.
So have we created a non-innovative sector, or have we learned from some of the initial mistakes we made? That's my point is the success.
It's a combination of
everything is easy to say after the failure.
You'll have to come back soon again to do Monday morning quarterbacking. The question on GBM and PANC. I know this is I know GBM is the stepchild of everything.
To be fair, I haven't worked much there.
But are you hearing anything though? You're not working on it because it's the whole industry is just not focused on it. But we need some help in the sector of GBM.
I don't think I can give you. No. I honestly
PANC.
There's some of the emerging KRAS data that I think is really interesting. I think some of the cancer vaccine data in post-adjuvant, very, very interesting.
Yeah.
I don't know if anything else I've seen.
No, it's a sad state in both of those tumors. Any thoughts, finishing thoughts or ending thoughts for us?
No, it's again, I wish as a sector we stop blaming companies for doing what the incentives are telling us to.
Yeah.
And we should embrace the fact that failure can occur, and a lot of these CEOs can get up and do something else again.
Yeah.
Why? It's a good thing. It's not a bad thing. And I'd rather fail fast and keep iterating.
The fail fast model is good.
Yeah. And look, the one thing I will say that I've been thinking about, which directly contradicts the thing I just said.
Oh, you just said for the last hour?
He's how did China build such a great biotech industry so quickly?
Yeah.
When they don't necessarily have the same pricing incentives that we have in the US market, which is probably an interesting question. And maybe there is a fair discussion to be had about, in order to approve another medication, how much clinical benefit does it have to get reimbursed? I think if you even tightened up that bar, it will have a negative impact on innovation, but maybe it forces these companies to be less iterative. If that's what you're seeking, I don't know if that's a good thing, because a lot of the innovation we're seeing are slight benefits, slightly better therapeutic window, slightly different toxin. And again, no one would have thought HER2 would have been the breakthrough it was, a toxin that everyone had given up on that was less potent. No one wanted less potent toxins with poor internalization, right? The whole point is it seems to work in areas where there's not even HER2 expressed. I just don't know how you can, what's the exact way to fix that? I think it's more again, incentives to allow earlier stage treatments, earlier stage intervention, and give them patent exclusivity in order to do that. To me, that's how we reshape our healthcare system to stop thinking in such a pathological extent once they have actually had disease, and we start looking a little more preventative. I think there is some real legislative, there is a room for the next Hatch-Waxman Act.
Yeah.
I think.
Yeah.
In order to create, again, a more preventative healthcare model for the US system. Not going to happen here.
So you bring up a good point, though. I know a lot of times we kind of feel why is America kind of bearing the cost of drug development, right? Now, all of the shifts you're seeing in China, I don't know the per capita spend in China versus the US, and I'm sure US is much higher in terms of drug delivery or drug discovery. But does the model then change? Because then maybe China is then taking on kind of the leapfrogging in terms of developing some of the drugs and carrying some of the cost as well. But then this population will benefit in the long run.
It's a really interesting question. Okay, a couple of thoughts there. Number one, it will be interesting to see if there isn't pricing and scale and access for the Chinese biotech industry. Will some of the innovation that the seeds were sown with that plan, the government plan from a decade ago, 15 years ago, will we see the same crop of companies in the 2030s?
Yeah.
Is TBD. I think there's actually a lot of US investors who are skeptical. They might think like this is the last, there's a wave of innovation. We may not have that before. I don't know. We'll see. But you asked an interesting, this is the question, like, is it a good thing that we do this or not? I would argue yes. Like I think it's an example of American exceptionalism. I think it's a good thing that we do this, that we spend, that unfortunately we are spending a lot on people's last year of life. No one else will do it. And if no one else does it, then that innovation completely dries up. Then they're right. I mean, you're just going to get put on palliative care. It's great. It's easy for us to say that until we're the ones doing it, and we're in a population that's going to continue to age. And let's be honest, we're also going to face severe population decline if you look at any of these developed countries. So we are going to have to care about. Frankly, that's probably where a lot of health, our resources should be devoted to. I know that's a controversial opinion. I think it's at least worth thinking about why. I think there's a lot of Americans who actually are okay that we spend as much on healthcare. Now, the interesting thing, like this is, I would argue, maybe more of a geopolitical discussion. There is probably a geopolitical advantage to being the center of innovation in the world, and you could argue that if, like, the China model was basically, we can't afford the price points that the US companies are charging for their drugs, so we're going to create our own companies and have a different price point because we have to serve 1.5 billion people. And that's actually probably the right thing for them. I think what will be interesting is, do those companies then start selling to India, to Russia, to Japan, right? Do Chinese biotechs not out-license assets to the US, but do they actually just start? And then what is the geopolitical power that you could probably have by doing that? The elephant in the room.
So we almost have like a first right of refusal at this time.
Well, when it comes to maybe branded drugs, yes. The thing no one wants to talk about is China's already there when it comes to generics. The FDA did a report. They can't even determine how much of our API or even precursors to API come from China. It's like 90%. And 9 out of every single 10 pills that are made are generic, that are administered by the US healthcare system. So, like, I don't know if we actually have the leverage here. That's not I don't think we do, actually. And the funny part is, why do you think all of the API development got put ex-US? Causes a lot of pollution. Actually,
makes sense.
to make API and precursor, it does cause a lot of pollution. So it got out licensed to China. So frankly, I don't know if we have as much leverage as we would think.
Also talk about that.
I think there should be, frankly, just more cooperation and the healthy balance that we're seeing. Again, I think the growth of the Chinese biotech industry has not just benefited the Chinese people, which is awesome. I think it's great. It's actually helped US companies develop drugs as well. So I think, again, we have to think more globally about where drugs are getting developed. And I think you're going to see, again, very good clinical sites all throughout the world, not just in the US.
That's great to hear. As you can see, like from our discussions for the hour and a half now, this is very exciting. And so we can go all day. Thank you so much for coming down. And then we've been planning it for a while. And thank you for making the time.
Thank you. Appreciate it.
Absolutely. We have to do this again. Thanks for joining in. I think the videos and the audios will be available soon. And send in your questions, and Akash, and we'll get back to you.